I want you to picture a trench in the middle of a war zone. It's muddy, it's chaotic, and there are two soldiers standing shoulder to shoulder. They're wearing the exact same uniform. They're holding the same rifle.
From a distance, they're identical.
Completely. To a general looking through binoculars a mile away, you literally cannot tell them apart. But if you could look inside their heads, you'd see two totally different realities. Soldier A is the ultimate sentinel, alert, scanning the horizon, ready to pull the trigger the second an enemy shows up. Soldier B,
he's checked out.
He's done. He's physically there, but mentally he has given up. His weapon is jammed, he's exhausted, and he's frankly just waiting for it to be over.
That is a really visceral image, but, biologically, that is the exact crisis our immune system faces when it's fighting something like cancer. We have these cellular identity crises.
Where two T cells look genetically almost identical, but one is a hero and the other is, well, useless.
Completely useless.
Yeah.
And that useless soldier is what scientists call exhausted.
And today, we're not just talking about identifying these soldiers, we are talking about hacking their minds. We're doing a deep dive into a paper published in Nature in 2026 titled Atlas Guided Discovery of Transcription Factors for T-Cell Programming.
This is a massive study. I really don't use that word lightly. The researchers essentially built a Google Maps for the genetics of the T-cell.
A Google Maps, okay.
Yeah, and their whole mission was to figure out exactly which genetic switches, we call them transcription factors, we need to flip to turn that exhausted soldier back into a fighter.
But without accidentally breaking the good soldier who's still on the job.
That's the critical part. Without breaking the good memory cells, it's a rescue mission on a molecular level.
It sounds like we have a lot to unpack. We need to get into this Google algorithm they used called Taiji.
Taiji, yeah. It's fascinating on its own. And we'll need to meet the villains of the story, these genes with names like Zscan20 and Jdp2.
Right, and then see how they use this other technique, PerturbSec, to actually prove this works in living tumors.
And just a heads up, we are going to get into the weeds of some genetic engineering here, but I promise sticking with it is worth it. The implications for cancer therapy are just huge.
All right, let's do it. Let's start at the root of the problem. We're talking about CD8 plus T cells. I've always heard them described as the assassins of the immune system.
That's a good way to put it.
But clearly in this paper, some of these assassins are falling asleep on the job.
Right. So in a normal, healthy immune response, say you get the flu, these CD8 plus T cells rush in, they kill all the infected cells, and then most of them die off. But a few remain. They stick around as memory cells.
The sentinels.
Exactly. And the paper focuses on a really specific type called TRM, tissue-resident memory T cells.
Tissue-resident. So that just means they don't float around in the blood. They pick a spot and they guard it.
You got it. They set up camp right there in the tissue, or in this case, the tumor. They are the good state. If a cancer patient has a lot of these TRM cells, their survival rates are generally much, much better.
Okay, so TRM is our alert soldier. Who's the burnout?
That would be the terminally exhausted T cell, or TEXterm.
TEXterm. It sounds like a bad sci-fi robot.
It kind of acts like one.
Yeah.
I mean, these cells are just dysfunctional.
Yeah.
They express super high levels of what we call inhibitory receptors. You might have heard of PD-1 or TIM-3.
Yeah, those are the targets for checkpoint blockade drugs, right?
The very same. These are basically off switches on the cell surface. And these TEXterm cells are just covered in them. They can't kill, they can't multiply, and worst of all, they're resistant to our best immunotherapy drugs.
So the goal seems simple. We want more TRM, more memory, and less TEXterm, less exhaustion. Why is that so hard? Why can't we just give them a drug that says, hey, stop being exhausted?
Because of what the paper calls the transcriptional overlap. This is the core paradox.
Okay.
If you look at the genetics of these two cells, the hero and the burnout, they look incredibly similar. They share key master regulators.
Give me an example.
Sure. There are these proteins, BLIMP1, BHLHE40, and NR4A2. These are crucial for the cell to function at all. Both the good memory cells and the bad exhausted cells have these turned on.
Ah, okay, now I see the problem. It's like trying to evict the couch potato twin from your house, but you accidentally kick out the marathon runner because they're both wearing the exact same face and the same t-shirt.
Huh. That is a surprisingly accurate analogy. Seriously. If you try to target the exhausted cell with, broad strokes like hitting one of those shared regulators, you might just destroy the memory cell by accident.
And if you kill the memory cells, you've lost your long-term protection. The cancer just comes back.
Exactly. So you need a sniper rifle, not a shotgun. You have to find the tiny specific differences between them.
And to do that, you need a map.
Precisely. You need to understand the whole network.
Which brings us to the methodology. This is where the whole Google thing comes in. The researchers built an atlas, but they didn't just count genes, did they?
No. Simply counting genes, what we call expression, it isn't enough. It's too noisy, Instead, they integrated 2 massive data sets across 9 different T-cell states. They looked at RNA-seq, which tells you what genes are being written.
Okay.
And they looked at ATAC-seq.
ATAC-seq. ATAC. That sounds aggressive.
It does, but don't worry about the full name. Just imagine you're walking down a hallway of hotel rooms. Some doors are open, some are locked shut. ATAC-seq basically tells you which doors are open.
And the doors are the parts of the DNA that can actually be used by the cell.
Exactly. If the chromatin is open, the cell can read the instructions inside. If it's closed, those instructions are archived away. So now they know which genes are active and which doors are open. But they still needed to know who was in charge.
And that's where the algorithm comes in, Taiji.
Taiji. It's a computational pipeline. And honestly, this is the coolest part of the whole study. It uses a personalized PageRank algorithm to find the bosses.
PageRank? Wait, as in the code Larry Page wrote for Google back in the 90s.
The very same logic. Think about how Google works. A website isn't ranked number 1 just because it has the most words on it.
Right, that would be useless. It's ranked number 1 because other important websites link to it.
Exactly. It's all about influence. Taiji applies this to biology. It looks at a transcription factor, let's just call it a project manager protein, and it asks, does this manager control other important genes? And are those genes themselves important managers? It builds this massive gene regulatory network.
So a transcription factor might not be very loud. It could have low expression. But if it's whispering orders to 50 other critical genes, Taiji flags it as a VIP.
You've absolutely nailed it. It calculates global influence. And this is how they solve that twin problem. By looking at the network, they could see that even though the twins looked the same on the surface, the bosses giving the orders underneath were actually different.
Okay, let's just pause for a second. This feels like a good time for a quick recap because that was a lot of tech.
Yeah.
Let me try to summarize. You tell me if I'm on track.
Go for it.
Okay, so point 1, we have these 2 T-cell states, memory, the good one, and exhausted, the bad one, and they look almost identical genetically.
Yep.
Point 2, because they share so many genes, it's really hard to treat one without accidentally hurting the other.
Right.
Point 3, the researchers used this Taiji algorithm, which acts like Google's PageRank, to map not just what genes are there, but who is influencing whom in the network.
That's a perfect summary. They moved from just a list of parts to a full-on wiring diagram.
So looking at this new wiring diagram, who are the villains?
This is where that Taiji analysis really paid off. They basically divided all the transcription factors into 2 buckets. Bucket 1, multi-state TFs. These are the generalists.
The ones that are active in both of the twins.
Right. They found genes like HIC1 and GFI1. These are shared. If you target them, you hit both the exhausted cell and the memory cell. You get collateral damage.
So we ignore those. We're looking for bucket number 2.
The second bucket is the single-state TS, the specialists. These are TFs that are uniquely active in only one state. And this is where they found the specific drivers of exhaustion. The villains are Zscan20 and Jdp2.
Zscan20 and Jdp2. Okay, I feel like we need a way to remember these names.
Well, think about what they do. They drive the cell into terminal exhaustion. They make the cell just stop working.
Okay, how about this? Zscan sounds like zero. Zero energy. Zero fight left.
I like that. Zscan means 0 energy. It works.
And on the flip side, who's the hero driving the good memory cells?
That would be KLF6. KLF6 is a unique regulator that only really shows up in those protective TRM cells.
KLF, keep living forever.
Keep living forever. It's scientifically a bit loose, but since memory cells provide that long-term protection, I'll allow it.
Great. So we have our cast of characters. Zscan20, 0 energy, is the villain. KLF6, keep living forever, is the hero. But this is where I get skeptical. I mean, computers predict things all the time. My weather app predicts rain, and it's sunny outside. Did they actually prove that Zscan20 causes exhaustion?
They did. And the way they proved it is just as impressive as the algorithm itself. They used a technique called perturb-seq.
Perturb-seq. Break that down for us. It sounds complicated.
It's a combination of CRISPR and single-cell sequencing. But a better way to think about it is like this. Imagine a massive fuse box that controls a whole building. You want to know which fuse controls the lights in the hallway.
Okay.
You could guess. Or you could systematically go and pull every single fuse one by one and see what goes dark.
So they're pulling fuses inside the T cells.
Exactly. They took thousands of T cells that are designed to fight a specific virus LCMV, which is a standard model for chronic infection. In each one of those cells, they used CRISPR to break or delete just one specific gene. Then they looked at each individual cell to see what happened to its identity.
So trial number 1. They pull the fuse for our villain, Zscan20. What happened?
The lights for exhaustion went out. When Zscan20 or Jdp2 were deleted, the cells, they just stopped becoming terminally exhausted. They didn't turn into those useless TEXterm cells.
But, and this is the million-dollar question, did they still become memory cells? Or did the whole cell just fizzle out?
That is the aha moment of the entire paper. They did still become memory cells. In fact, they were even better at it. This proved what they called the decoupling hypothesis.
Decoupling, meaning the road to exhaustion and the road to memory are two totally separate highways.
Exactly. For years, we worried they were inextricably linked, that you couldn't get rid of one without the other. This proved you can cut the wire to exhaustion without touching the wire to memory.
Wow. That is huge. And just to prove the point, what happened when they pulled the fuse for one of those shared genes like HIC1?
Just what they were afraid of. When they deleted HIC1, yeah, exhaustion went down a bit. But the memory cells were also destroyed. It just confirmed that specificity is everything. You can't just carpet bomb the genome.
You have to hit the right target.
Precisely.
And what about our hero, KLF6? Did they do anything with that one?
With KLF6, they did the opposite. Instead of pulling the fuse, they rewired it to be always on. They overexpressed it.
A kind of super soldier serum.
Essentially, yeah. And the result was this massive expansion of TRM cells. Specifically in the small intestine, they saw a 15-fold enrichment. They basically created an army of super sentinels that wouldn't burn out.
Okay, I'm getting a really clear picture now. Let's do another quick recap because we're about to shift gears into the clinic.
Sounds good.
1. The Taiji algorithm found the unique drivers. 2. Zscan20 drives the bad stuff, KLF6 drives the good stuff. And 3. Using that fuse box method, or TURBSEC, they proved that if you kill Zscan20, you stop the burnout, but you keep the memory.
You've got it.
But, you know, mice are not humans, and a virus isn't cancer. Does any of this actually work on a real tumor?
That's the final frontier of the paper. And yes, they went there. They moved to a melanoma model, so skin cancer in mice. And they used a therapy called adoptive cell transfer.
Which means?
You take T cells out of the body, you edit them in the lab, and then you infuse them back into the patient, or in this case, the mouse, to go fight the tumor.
So they took these Zscan20 deleted cells, the ones with the exhaustion fuse pulled, and put them up against a tumor.
And the tumor shrank significantly. The cells that had Zscan20 deleted didn't give up. They stayed in the fight, transitioning into these progenitor-exhausted or effector states. They kept working.
But they didn't stop there. They did a combo move.
They did. They combined the Zscan20 knockout with anti-PD1 therapy.
Anti-PD1, that's the standard immunotherapy drug we use now. It basically takes the brakes off the immune system.
Right. Think of PD1 as an external brake pedal that's on the cell surface. And think of Zscan20 as the internal engine code that's telling the cell to slow down.
I see.
So when they removed both using the drug for the external brake and CRISPR for that internal code, they saw significant tumor regression and much better survival than doing either one alone.
One releases the parking brake, the other fixes the engine.
That is a perfect analogy. The synergy was really powerful.
Now for the question I know everyone listening is asking, does this code exist in my cells? Or is this just some weird mouse thing?
They checked. They looked at a human pan-cancer atlas data from glioblastoma, from renal cell carcinoma, and others. And the good news is the villains are conserved.
Conserved?
It means evolution kept them. The same transcription factors, Jdp2, Zscan20, were active in human exhausted T cells, the same ones.
So the code is the same?
It's remarkably similar. They even did a validation experiment in human T cells in a dish. When they deleted Zscan20 or Jdp2 in the human cells, they saw an increase in stem-like markers, which is good, and a reduction in those inhibitory signals. It strongly suggests this strategy could actually work in human immunotherapy.
This feels revolutionary. It feels like we are moving from discovering biology to actually engineering it.
That is exactly the shift we're seeing. So what here isn't just about finding Zscan20, it's about the platform. We can now use algorithms like Taiji to generate these precise recipes for how we want a cell to behave.
We can decouple the bad traits from the good traits.
Yes. We are no longer guessing. We can say, okay, I want a cell that remembers the tumor, so let's add KLF6. I want it to not get exhausted, so let's delete Zscan20. And I want it to ignore the tumor's defense signals, so let's add an anti-PD-1 drug.
It's customized medicine, but on a genetic level. So let's bring it all home. What are the key takeaways from this deep dive?
If I had to boil it all down, first, the Taiji algorithm lets us see beyond simple gene expression and map the actual influence networks, the bosses of the cell.
Right.
Second, using that map, we identified Zscan20 and Jdp2 as these very specific drivers of exhaustion and KLF6 as a driver of memory.
The villains and the hero.
Exactly. Third, we proved we can decouple these states. We could delete the bad without losing the good. And finally, combining this kind of genetic engineering with current drugs leads to much better tumor control.
It's a masterclass in how computation and biology are merging.
It really is. And it raises a question I've been thinking about ever since I read this paper.
Oh yeah, let's hear it.
If we can now edit the operating system of a T cell to remove the code for exhaustion, what other cellular codes could we rewrite?
Oh, that's interesting.
I mean, think about it. Exhaustion is just a state. Aging is a state. Autoimmunity, where your body attacks itself, is a state. If we can use Taiji to map the specific single-state drivers of those conditions, could we edit cells to never age?
Wow.
Could we edit immune cells in a person with type 1 diabetes to simply ignore the pancreas?
You're saying we could theoretically delete the code for getting old or attacking yourself without breaking the rest of the cell?
If this decoupling hypothesis holds true for other systems, why not? We just need to find the right transcription factors. The map is the hard part, and Taiji just gave us a powerful new way to build the map.
That is a staggering thought to end on. We're not just reading the book of life anymore, we're starting to edit the typos.
And hopefully write a better ending.
If you want to see the atlas they built, the visual abstract is really stunning. Definitely check the show notes. It's a beautiful example of data visualization. Thanks for joining us on this deep dive into the T-cell operating system. Stay curious, and we'll catch you on the next one.